We are investigating how gravity affects the growth direction and rates of newly germinated seedlings by observing roots and shoots in a controlled setup. Gravitropism is a growth response to gravity in which roots show positive gravitropism by growing toward gravity and shoots show negative gravitropism by growing away from gravity. Plants sense gravity when dense statoliths settle in specialized cells, and this changes oxygen transport so that different growth on the upper and lower sides causes bedding. Our independent variable is the gravity condition or orientation. Our dependent variables are the angle of curvature and the change in length of roots and shoots. And our control variables include seed species and age, temperature, moisture, oxygen, and exposure to light. The equipment is fast germinating seeds, petri dishes with moist filter paper, a fine marker grid, a clinostat or slow rotating turntable to cancel a constant gravity vector, a ruler and protractor or camera for measurement, a dark box, and a timer. First, we soak the seeds as required, line each dish with moist filter paper, draw a simple grid on the lid, and mark an arrow showing the gravity direction. We place equal numbers of seeds evenly spaced with the seed hilum aligned sideways where possible and label the dishes for the planned conditions. One dish is kept vertical and stationary, one dish is kept horizontal and stationary, and one dish is mounted on a clinostat rotating slowly at about 1 revolution per minute to cancel a stable gravity direction. We incubate the dishes for a fixed period such as 24 to 48 hours in the dark to remove phototropism, and we keep temperature and moisture constant. At the end, we photograph or trace the seedlings, measure root and shoot lengths, and measure the angle of each organ relative to the gravity arrow or to the initial orientation. We calculate the mean curvature angle and mean growth rate for each condition and plot curvature or angle against condition, expecting the stationary dishes to show roots bending downward and shoots bending upward, while the clinostat shows little or no curvature. To improve reliability, we use many seeds per treatment, randomize their positions, keep rotation speed constant, maintain moist paper, and use seeds of the same batch and age.